Hello guys, so welcome to my channel. So as you know, we studied about this protein folding and protein misfolding, how the protein is synthesized in the ribosome, and sometime instead of folding in the native formation or conformation, it can be misfolded, right? So we'll talk about what this misfolding will lead to, what kind of diseases. So So as we studied, like there are many processes to fold the protein, and despite all this processes, there are misfolding does occur, and like the in natural reaction, like if the protein is misfolded, it is like degraded, or like it's done by an autophagy, or there is another process known as ubiquitin proteasome system process. So all these kinds of process are there in our body, so that will remove the misfolded protein. But many conditions are associated with this misfolding of protein, such as type 2 diabetes, Alzheimer disease, Huntington disease, Parkinson disease, right? So what happens in this? So there, if the a soluble protein, right, is normally secreted from the cell, is secreted in a misfolded state and converted into an insoluble extracellular amyloid fiber. So if the protein is secreted from the cell and it's instead of folded state, it is misfolded, then it will be converted into an extracellular amyloid fiber, and that diseases are collectively referred to as an amyloidosis, right? That refers to as amyloidosis. Right? This is E here. So the fibers, the amyloid fiber, are highly ordered and unbranched with the diameter of, what's the diameter of amyloid fiber? It is 7 to 10 nanometers, right? And a high degree of beta sheet structure. So let's say, for example, all right, so we'll draw it here on this side. So for example, I'm just drawing a native protein. All right, so we'll just draw a native protein. Like for example, this is a kind of a native protein, right? That's in the folded state. And it's connected. Right, so this is the one kind of native protein, and this native protein has a region in this protein with the beta sheet. Right, it has a region of beta sheet that partially goes, that includes that, like when it's processed, for example, in the cell, it goes partially folding, it's not fully folded. So in what happen if this is, for example, this protein goes, instead of going into a full conformation or full folding, it just goes into partial folding. So like something like this. Let's draw a random diagram. This, like this will happen here, and this will have here. All right, something like this. So this is misfolded protein. So what will happen if this beta sheet in the small number of molecules, before the protein goes complete folding, right? The beta sheet region of one polypeptide associate with the same, it will associate, what it will do, it will associate with the same region in the another polypeptide, forming a nucleus. So this is a kind of a nucleus of an amyloid. So this will form a nucleus of amyloid. So normally if this happens, like it should go into a denaturation state, like something like this, and it will be denaturated. It should be denaturated, and that's a definition, three-dimensional substitution to loss of function, right? So it will be denaturated, right? But and maybe it will be degraded by one of these methods, right? So with this misfolding, so what will happen if one, suppose one protein goes like this? Sometimes what will happen? So for example, this is here, here, right? Okay, you can see that, right? And if this is like this and like this, and this will uh, it will go like this again, and it will have like this, right? And this will be attached here. Right, so what happens, like in this misfolded state, sometimes additional protein molecules slowly associated with this amyloid and extend it to form an amyloid fibril. So this will form amyloid fibril, like additional protein molecules start associated with this amyloid, this misfolded protein, right? Right, and when this amyloid fibrils are together, it will form amyloid fibers, right? In the sum of the amyloid fibers, the overall structure includes two layer of beta sheet, such as like there is amyloid beta peptide that will have two layers of beta sheet, and many proteins can take on the amyloid fibril structure as an alternative to the normal folded conformation, and most of this protein have a concentration of aromatic amino acid residues. So like sometime when this proteins, for example, some protein goes to this structure, this kind of amyloid fibril structure, so many protein can take on just this kind of structure only, and instead of their normal conformation, folded conformation, and most of this protein which has which develop this kind of structure, amyloid fibril structure, will have a concentration of aromatic. So they will have aromatic amino acid residue. For amino acid residues, right, in the core region of beta sheet or alpha helix, whichever they have, right? So it will have aromatic amino acid, and so what happens, the this amyloid fibril grows in the extracellular space. It will start growing in the extracellular space, and other parts of the protein that fold differently remaining on the outside of the beta sheet core in the growing fibril. So other parts of the protein that are not here in the core, that will be outside on the beta sheet core in the growing fibril. So the effect of aromatic residue in stabilizing the structure. And so like it's not like all the protein that are coming outside will just go in the misfolded state, like because most of the protein molecules that will come will fold normally. So that's the reason the onset of symptoms of the amyloidosis is often very slow. But if a person inherits in the mutation, such as a substitutions with this aromatic residue at a position that favors formation of amyloid fibrils, this disease symptom may begin at an early age. Right, so in eukaryotes, the protein destined for the secretion undergoes its synthesis or its initial folding in the endoplasmic reticulum, right? In the ER. That's an endoplasmic reticulum. So the protein that is destined for the secretion undergoes initial folding in the endoplasmic reticulum. So when the stress condition arises, like when there is more need of protein, so the what will this ER will? The protein synthesis threatens to overwhelm the protein capacity of the ER. Right? So when there is like a stress situation, stress condition arise, ER has a limited folding capacity, right? So when there is like so many folded proteins started coming, so the folding capacity of ER will be limited, and the unfolded proteins started accumulating. What will happen in the stress condition? When proteins started coming, unfolded protein will start accumulate, right? So if the unfolded this if this unfolded protein start accumulating, then there is a condition or there is a trigger That trigger is known as UPR. So that means unfolded protein response will start. So our cell has some mechanism, like if this things happen, like there should be, like for example, if you are doing some work and if you are overloaded with work, so like your manager or like whoever is your supervisor, they will give you extra hand so that the pending work can be done. So that's the same situation like here as well. So if the unfolded proteins start accumulating, so there is unfolded protein response triggers, and this triggers, there are set of transcriptional regulators that constitute this UPR, brings various systems in alignment by increasing the concentration. So for example, what it will do, it will increase the concentration of this saporins, right? This saporin. So this saporin, what it does, it folds the protein, right? So that's one of the help, like it will increase the concentration of saporins. Then, or what it will do, it will decrease the rate of the protein synthesis, like from where the protein is coming, and it will say, okay, that's it, I think we have to stop the protein synthesis or decrease the protein synthesis. So all this unfolded protein first should be folded, right? And or else, so these are some of the mechanism. And if amyloid aggregates that form before the UPR system can come into play, may be removed. So if suppose this unfolded protein goes into this amyloid fibrils or amyloid, or it start accumulate the amyloid aggregate, it can be degraded by autophagy as well. This can be degraded by autophagy. So what is autophagy? That's a process that the aggregates that are first encapsulated in a membrane or a vesicle, then this content of the resulting vesicle are degraded after the vesicle docks with the cytosolic lysosome, right? And there is another method beside this autophagy. This misfolded proteins can be degraded by a system, that is system of proteases. So that's an enzyme, and that system is known as ubiquitin protease system. So that's the another system for the degradation of protein. So defect in any of these systems will decrease the capacity to deal with this misfolded proteins and the increased propensity for the development of amyloid related disease. So these are so many mechanisms, and if any of this mechanism fail, then there is a threat for this amyloid related disease. So that's why this UPR is a complex response involving many protein factors and signals, and inactivation of this UPR response component may have a positive or negative effects on the degree of protein misfolding. So that's why this system is an attractive drug target for the protein misfolding diseases, that's like especially amyloid diseases. So the pharmaceuticals that are making drugs, they are trying to make a drug that target this UPR system, right? So now if for example amyloidosis occurs, so there are different kinds of amyloidosis. So some amyloidoses are systemic, involving many tissues. The one is primary systemic amyloidosis. So it's all right. So this is one. So in this primary systemic amyloidosis, this is caused by deposition of fibrils consisting of, so how this is caused by deposition of fibrils. Of course it will be the fibrils, but what kind of amyloid fibrils it has? Fibrils, what kind of fibrils? Consisting of misfolded immunoglobulin light chain. So this will contain light chain. Right, so this primary systemic amyloidosis is caused by this deposition of fibrils, this amyloid fibrils, that will have misfolded immunoglobulin light chains or a fragment of light chain that is derived from the proteolytic degradation. Or what will have, it will have, instead of this deposition of fibrils, it can also have fragments of light, this fragments of light chain that is derived from this proteolytic degradation. So for example, this protein is degraded, but like there are some fragments that are left that can also cause this primary systemic amyloidosis. So the mean age of onset is about 65 years for this primary systemic amyloidosis, and patients may have symptoms like fatigue, hoarseness, swelling, and weight loss, and many die within the first year after diagnosis. And sometimes the kidneys or heart are often the most affected organs. There are some amyloidosis that are associated with other types of diseases. Like for example, people with certain chronic infectious or inflammatory diseases such as rheumatoid arthritis, tuberculosis, cystic fibrosis, and some cancer can experience a sharp increase in the secretion of an that in and all this infection or inflammatory diseases that people might have, they can experience a sharp increase in a secretion of an amyloid prone peptide. Amyloid prone peptide. So what is called the amyloid prone peptide, called as a serum amyloid A or SAA protein. Right? So this is a protein. Right. So the peoples that have infectious diseases or inflammatory diseases, especially like rheumatoid arthritis, tuberculosis, cystic fibrosis, and some cancer, can experience a sharp increase in the secretion of this amyloid prone peptide that is called amyloid serum amyloid A, SAA protein. So this protein or fragment of it deposits in the connective tissue of the spleen. So this protein, all right, this protein, where it goes, it deposits in the connective tissue. Deposits in the connective tissue of what kind of organs? Spleen, kidney, liver. So maybe I will write it down here. Spleen, liver. Oops, there's no space. Spleen, liver, kidney, and around the heart. So people with this condition is called secondary systemic amyloidosis. So I will write it down here. Secondary systemic amyloidosis. All right. So what the primary has, they have a fibril of this misfolded immunoglobulin light chain or the fragments of it from the proteolytic degradation. Or for the secondary systemic amyloidosis, they have this kind of prone peptide, that is serum amyloid A or SSA protein, and that is deposited in the connective tissues of like heart or kidney, liver, and the disease is generally fatal and people may die within a few years. So more than 80 amyloidoses are associated with mutation in the transthyretin. Right, so this is two types. There is another type. So there is a protein known as, I think you can see, right? Transthyretin. So this protein, what it does, that binds to and transport thyroid hormones from distributing them throughout the body and brain. So transthyretin, what is that protein what that binds to and transport the thyroid hormone, that this will bind to the thyroid hormones, and it will transport the thyroid hormones and distribute them throughout the body and brain. So there are more than 80 mutations or 80 amyloidoses are associated with this with mutation in this transthyretin, like more than 80 amyloidoses just with this protein, right? And a variety of mutations in this protein lead to amyloid deposition concentrated around different tissues, thus producing different symptoms. So this protein is also right related to this amyloidosis, but with different symptoms. So amyloidoses are associated with inherited mutations in the proteins such as lysosomes, fibrinogen A, alpha chain, apolipoprotein A1 and A2, right? All of this kind of proteins that can also show different kinds of amyloidosis. So there, but there are some other kind of amyloidosis that are just associated with particular organs. So the amyloid prone protein is generally secreted only by the affected tissue, right? The amyloid prone protein, what that the is generally secreted by the affected tissue, and its locally high concentration leads to amyloid deposition around that tissue, right? So that because of that reason, it can occur in a particular organ as well, right? So there, the one common site of amyloid deposition is near the pancreatic islet B cell. All right, so we'll do like this. Right? Let's separate pancreatic islet beta cells. So this area has a that's a common site of the amyloid deposition, pancreatic islet beta cells. These cells are responsible for insulin secretion and regulation of glucose metabolism. So for example, if there is so much of glucose, it will secrete insulin and that will maintain the glucose level in our body. So this is the pancreatic islet beta cell is the one of the common site for amyloid deposition, right? So what happens? The secretion by beta cells of a small peptide. So this beta cell will start secreting a small peptide that is has just 37 amino acids. 37, I think, okay, you can see 37 amino acid peptide that is called yeah, I'll write it down. That's what it's called. It's called islet amyloid peptide. Amyloid peptide or IAPP. IAPP, or it's also called amylin, right? And this can lead to amyloid deposit around these islets, gradually destroying the cells, right? If these beta cells start secreting a small peptide that is called this islet amyloid polypeptide, right, or IAP, or also known as amylin, can lead to amyloid deposit. So this is polypeptide, eh? It's not just a peptide because it has 37 amino acids, right? So can lead to amyloid deposition around these islets and gradually destroying the cell. But the healthy human has 1 to 1.5 million pancreatic beta cells. How many it has? 1 to 1.5 million. 1 to 1.5 million beta cells. So with progressive loss of these cells, glucose homeostasis is affected, and eventually when the 50% or more of these cells are lost, the condition matures into type 2 diabetes mellitus. Right, so that forms type 2, or we don't have space, I'll write it down here. So it will develop type 2 DM, diabetes mellitus, right? Alright, so that causes type 2 DM. Okay, so I will just remove, so we'll have some more space because there is more as well for amyloidosis. All right, I will leave this. that will separate from this. All right. Okay, so this is one of the localized amyloidosis. So there are amyloidosis deposition disease that triggers the neurodegeneration, particularly in older adults. There are special class of localized amino acids, right? So what it does, it can also develop neurodegeneration, amyloid deposits. So special case is the Alzheimer disease, right? Alzheimer disease, right? This Alzheimer disease is associated with extracellular amyloid deposition by neurons involving the what it involving the amyloid beta peptide. Amyloid beta peptide. And this amyloid beta peptide is derived from a larger transmembrane protein. What's that protein? That's all right, I will write it down. Derived from larger transmembrane protein, amyloid beta precursor protein. Beta precursor protein. So this protein is found in most of the human tissues, right? So in the Alzheimer disease, that's associated with extracellular amyloid deposition by neurons that will involve the amyloid beta peptide. And you can see the picture of amyloid beta peptide. That's just a computerized picture that is shown here, right? And this amyloid beta peptide is derived from a larger transmembrane protein. It is a transmembrane protein, remember that, right? Transmembrane, so it's on the membrane. That's amyloid beta precursor protein, and this is found in most of the human tissues. So when this large when this protein is a part of a larger protein, so the peptide that composed of a two alpha helical segment. So this protein has alpha helical segment as well, two alpha helical segment that is spanning the membrane, right? It's inserted into the membrane. So when the external and the internal domains are splitted off by the specific proteases, that's an enzyme, the relatively unstable amyloid beta peptide leaves the membrane and loses its alpha helical structure. So what happens after that? It can then take the form of two layers of extended parallel beta sheet. So after losing the alpha helical structure, what it can do? It can take the form of two layers of extended parallel beta sheet, which can slowly assemble into the amyloid fibrils. Right, so you can see on your screen. So this is a computerized picture image for the amyloid fibrils. So it can be something like this. After losing the structure, alpha helical structure, it can be arranged like this. That's a beta sheet in parallel to each other, and that will form amyloid fibrils. And deposit of this amyloid fibrils is thought to be the primary cause of Alzheimer's disease. But a second type of amyloid-like aggregation involving a protein called tau. So, but there is also a second type of amyloid-like aggregation that's involving what kind of protein is that? That is tau, right? Also occurs intracellularly or in neurons in people with Alzheimer disease. But most of this tau protein, okay, so I will write it down here, protein, so you will know that protein. So this protein cause of frontotemporal dementia. Dementia and Parkinsonism. Right? So what we studied about this Alzheimer disease? So it has this amyloid beta peptide, right? that is derived from this transmembrane protein that's also known as amyloid beta precursor protein, right? And when this transmembrane protein is a part of a larger protein and it has the alpha 2 alpha helical structure, and if it's like splitted by the enzyme proteases, it will be unstable and it will, what is the amyloid beta peptide will leave the membrane and it will lose the alpha helical structure, but it can then take the form of two parallel beta sheet which can slowly assemble into the amyloid fibrils, and deposit of that amyloid fibrils can lead to this Alzheimer disease. So that is seems to be the primary cause of Alzheimer disease. But there is a second type of protein that is called a tau. So this protein is intracellular in neurons with found in the people with Alzheimer's, right? Right. So if this tau protein is mutated and if it's inherited, so I will write it down here, inherited mutation, right? So the inherited mutation of this tau protein will results it will not result in Alzheimer disease, but it will result in the frontotemporal dementia and Parkinsonism, a condition or the symptoms. This is a condition with symptoms resembling Parkinson disease that can be equally devastating. So there are several other neurodegenerative conditions that involve intracellular aggregation of misfolded proteins. So for example, in Parkinson disease, the misfolded form of the protein alpha sy so what this will have alpha synuclein. So this alpha synuclein, right? This is a protein that aggregates into a spherical filamentous masses. So this, okay, I'll write it down. This alpha synuclein protein, what it does, it aggregates into a spherical, not something like beta, parallel beta sheet, but into a spherical filamentous masses. As filamentous masses called, what this is called? This is called Lewy bodies, right? Lewy bodies. So in Parkinson disease, what happens? This involves misfolded form of the protein alpha synuclein. So that aggregates into spherical filamentous masses that is called Lewy bodies. There is another kind that is Huntington disease. So in this Huntington disease, that will involve the protein Huntington. So that will involve protein Huntington, which has a long polyglutamine repeat. So this protein has a long polyglutamine repeat, and in some individuals, the polyglutamine repeat is longer than the normal, and a more subtle type of intracellular aggregation occurs. So the Huntington disease involves this kind of protein, Huntington protein. So what the researchers did when the mutant proteins that is involved in the Parkinson's and the Huntington disease, if these are expressed in the Drosophila melanogaster, that's a fly that display this. If it's expressed in that fly, it displays neurodegeneration uh, expressed as eye deterioration or tremors and early death. So all of these symptoms are highly suppressed if expression of HSP70 chaperone is also increased. So if you remember chaperones, right? So HSP70, that's a one kind of chaperone that folds the protein. So if this is increased, the symptoms are highly suppressed. But the protein, so it's not always like if the protein is misfolded, it should cause amyloid disease. It's not the case. Or it's not the amyloid formation to cause this serious disease. For example, cystic fibrosis. Right, I will do a star. So this will not cause amyloid disease. Cystic fibrosis. So this cystic fibrosis is caused by the defects in the membrane bound protein. That's all right. So I will write here, caused by defect in membrane bound protein. So this causes a defect in the membrane bound protein. What's called that protein? Called as cystic fibrosis. Cystic fibrosis. All right. So cystic fibrosis, transmembrane. So it's called cystic fibrosis, transmembrane conductance. Conductance regulator, or shortly it's known as CFTR. Right, so this cystic fibrosis, this is caused by the defect in the membrane bound protein. So this is not, so this is a membrane bound protein that's called a cystic fibrosis, transmembrane conductance regulator. So which this protein acts as a channel for chloride ion. So this protein act as a channel for chloride ion. So the most common cystic fibrosis, so causing mutation in the deletion of PHE residues. So this protein is associated with this cystic fibrosis, and what happens in this protein? So there is a mutation. Okay, so I'll write it down here. So there is a mutation. In that mutation, that causes a deletion. What it happens in this mutation? Deletion of PHE. That's residue. PHE is the phenylalanine amino acid residue at the position of 508. Right? Deletion of PHE residue at the position of 508. At the position of 508 in this CFTR, which causes improper protein folding. So what happens in this cystic fibrosis of the mutation of this protein? If there is a deletion of the PHE, that's phenylalanine. I'll write it down here. PHE is phenylalanine. That's amino acid residues at 508 position in the CFTR that will cause improper protein folding. So most of this protein is then degraded and its normal function is lost, right? So if this protein is mutated, so all of this protein is degraded and its normal function is lost, and many of the disease related mutation in the collagen also cause defective folding. All right, so that was cystic fibrosis. There is one more thing for the misfolded protein, and that disease is really fatal. So, right, so we'll just remove this and we'll just discuss a small part of it. It's a historical thing. All right. So the misfolded brain protein seems to be the causative agent of several rare degenerative brain diseases in mammals, right? So there, if there is any misfolding in the of the brain protein, that will cause several degenerative diseases, brain disease. Perhaps the best known of this is, so the best known is The best known brain diseases: bovine, spongiform, and spongiform encephalopathy, right? Or it's also known as mad cow disease, right? Or mad DIS. I will just do mad cow disease is also known as mad cow disease, and related diseases include. So this has a related disease as well, right? So I will do this. Related disease includes Kuru, and there is uh, Creutzfeldt-Jakob disease, right? Jacob disease. So this are in humans, right? This both are in humans. In humans. In sheep, right? There is scrapie. It's called in sheep. This is goes in the sheep. Or chronic wasting. This is in deer and elk. So this bovine spongiform encephalopathies. So this disease is also referred to as a spongiform encephalopathies because it has so many diseases, and because the disease brain frequently becomes riddled with holes. If you see in the microscope, it would show riddled with holes. So the progressive deterioration of the brain leads to a spectrum of these neurological symptoms. So this will have like weight loss, erratic behavior, problems with posture, balance, and coordination, and loss of cognitive function. And these diseases are fatal, right? So so there are like so many from 1960s, like investigator were trying to find the causative agent, but the infectious agent, this has been traced to a single protein. They believed that it was a nucleic acid, but the infectious agent of this is a single protein. And all right, so okay, well, the infectious agent is believed to be a single protein, which, so this has been named as prion by Stanley. Single protein, that is prion, and this is named by Stanley Prusiner. So the name was derived from the proteinaceous infectious, and this prion protein is this prion protein is a normal constituent of brain tissue in all the mammals. So its role is not known in detail, but it may have a molecular signaling function. So when the strains of mice lacking the gene of this PRP, it's also actually wrote like this PRPC for the cellular prion protein, right? If the mice is, it's saying that this mice is lacking this PRP, that suffers no obvious ill effects, but the illness occurs only when the normal cellular PRP, this PRP that's cellular, occurs in an altered conformation as PRPSC. So SC is denotes scrapie. SC equal to scrapie, right? So when the symptoms occur, when this cellular prion is transformed to this conformation, scrapie prion. So the structure of the PRP, the cellular prion has two alpha helices. So on your screen you can see the image of this. So it has a two alpha helices. So the structure of, but the scrapie PRPSC is very different, which much of the structure converted into, as you can see this, they converted into the amyloid beta sheets. So the interaction of the scrapie prion with the cellular prion converts the later to also a scrapie prion. So initiating a dominion or a domino effect in which more and more of the brain proteins convert to the disease causing form. So the mechanism by which the presence of the scrapie prion leads to spongiform encephalopathy is not understood, but right, how it forms this spongiform encephalopathy. But the inherited forms of the prion disease, a mutation in the gene encoding prion produced the changes in one amino acid residues that is believed to make the conversion of this cellular prion to the scrapie prion more likely. But a complete understanding of prion disease awaits new information on how prion protein affects brain functions. Right. The significance of prion may extend well beyond spongiform encephalopathies. Might be evidence is building that the prion-like proteins may be responsible for additional neurodegenerative diseases such as multiple system atrophy (MSA), a disease that resembles Parkinson disease. That was all related to the misfolding of protein. Right. So that we finishes our structure of protein and related pathologies that happens if protein is not folded properly. So next we'll study about the protein function. All right, thank you guys for watching and don't forget to subscribe to the channel.